Welcome to the second part of our high temperature stress lecture. In this part, we'll be covering resistance. We'll be comparing tolerance versus avoidance. We will have a look at heat shock response, its characteristics, its regulation, and signal transduction pathway. After that, we'll talk about heat shock proteins, their signal transduction pathway, their functions, and the HSP families or heat shock protein families. Lastly, we will talk about the non-heat shock proteins, their signal transduction pathway, and their functions. Plant resistance to heat stress can be viewed as tolerance or avoidance. Avoidance is when the plant does not come to equilibrium with its environment with respect to the stress. An example on that is the evaporative cooling, that is a significant way for plants to dissipate solar energy. Tolerance, on the other hand, involves coming to equilibrium with the stress, but the plant is able to avoid, reduce, or repair injury. Tolerance includes constitutive adaptations as well as acclimation responses. The most studied acclimation process is the heat shock response, but there are additional modes as well. Our working definition of the heat shock response is follows. When organisms are exposed to increases in temperature, there is a rapid, large-scale synthesis of a group of stress response proteins called heat shock proteins or HSP. Heat shock proteins play various roles in thermotolerance. The heat shock response is very widespread taxonomically. It was discovered in Drosophila but has subsequently been detected in bacteria, insects, vertebrates including mammalian cell cultures, and higher plants. It has been found in all organisms checked so far except perhaps for 2 species of Hydra. The response is very rapid, with heat shock proteins constituting a major part of protein synthesis within 30 minutes after soybean seedlings are shifted from 30 degrees Celsius to 40 degrees Celsius. Other groups of heat shock proteins can show up later, but the bulk are fast. As a measure of how extensive production can be, heat shock mRNAs can accumulate to about 20,000 copies per cell in 2 hours. During heat shock, the normal pattern of protein synthesis slows or stops, with a shift from housekeeping proteins to heat shock proteins. Heat shock proteins transiently accumulate for 1 to 4 hours depending on the temperature, then levels decline. Regulation of heat shock genes in prokaryotes involve RNA polymerase sigma factor control. Sigma factors are subunits of RNA polymerase that recognize promoter regions in DNA molecules. Since there is more than one sigma factor, RNA polymerase can recognize different promoter sequences. Heat shock triggers an increase in sigma 32, which causes a shift to heat shock promoters. In eukaryotes, regulation of heat shock response is more complicated due in part to transcription and translation being physically and temporally repeated. There are 2 main ideas of how the heat shock is sensed by the plant. One school of thought is that changes in membrane viscosity trigger a signal transduction pathway. As the temperature increases, membrane entropy increases, leading to a mechanical signal analogous to the feedback loop proposed for low temperature exposure. The other idea involves disruptions of protein equilibrium, with temperature-induced changes in structure being a key. The heat shock is likely to produce a large increase in partially unfolded proteins and a corresponding decrease in free molecular chaperones. We will develop this model further once we explore the heat shock transcription factors. The number of transcription factors identified in model species runs at about 20 to 30. Transcription factors are either constitutively produced and inactive, or not present in the absence of heat stress. Activation involves polymerization to a dimer or a trimer, localization of the nucleus, and binding with heat shock elements and promoters of heat shock genes. A number of structural components of heat shock transcription factors have been identified. A DNA binding domain containing a helix-turn helix motif binds to the heat shock element of the promoter. Polymerization domains, either dimer or trimer, involve in hydrophobic patches of leucine zippers. Nuclear localization signals are logical, and activator domains interact directly or indirectly with the transcription initiation complex. In the next group of heat stress resistant slides, we will examine functions of heat shock gene products. The products of heat shock genes, the heat shock proteins, function as molecular chaperones through their capacity to bind to proteins. This role is important in many aspects of protein homeostasis. Therefore, it is not surprising that molecular chaperones are also active in non-stressed cells. Molecular chaperones play a major role in protein folding, protein transport, regulation of activity, and prevention of aggregation. The heat shock families are classified after their molecular weight into 5 families: the HSP100 family, the HSP90 family, the HSP70 family, the HSP60 family, and the small HSPs. The timing and location of protein folding is crucial to achieving a functional 3D structure at the correct location. The hierarchical model of protein folding uses an analogy to building blocks. Sections can establish secondary structure, then come together to form tertiary structure. The folding process is governed by changes in free energy. A fully folded protein has the lowest overall energy in an optimal environment. However, a fully unfolded protein has the greatest entropy. Therefore, the stability of folded proteins is a balance between the decrease in free energy achieved through interactions such as hydrophobic interactions and intermolecular bonds, and the increase in free energy resulting from decreasing entropy. As a result, many proteins are marginally stable and can become denatured by changes in the environment. Some proteins can go through the folding process and achieve a low free energy state without help. Other times, assistance from helper molecules, including molecular chaperones, is required. Molecular chaperones are proteins that can assist in the folding process. They promote proper folding of polypeptides but do not direct or create the pattern. One way to look at it is they prevent incorrect patterns or provide an energetic push. Different types of molecular chaperones fill different roles, with the binding role shown in the figure. When a protein is being made, it emerges from the ribosome and develops some secondary structure. Hydrophobic stretches are exposed, which is energetically unfavorable. So there is a tendency to group them together. This initial pattern of grouping may or may not be the correct orientation. Exposed patches can also stick to hydrophobic patches of other molecules, leading to aggregation. Chaperones bind to hydrophobic stretches. This prevents improper interactions until the correct conformation is achieved. At this point, there is a controlled release of chaperones. The molecular chaperone mechanism for the HSP70 family is shown. Chaperone activity takes place while the polypeptide is attached to the ribosome. Unfolded polypeptides binds HSP40, a co-chaperone. Then the HSP70 binds to the polypeptide. It has an ATP attached. The ATP is hydrolyzed to ADP, and then a complex releases HSP40, which is then recycled. When the chain is long enough, the ADP is exchanged for ATP. This involves nucleotide exchange factor, GRPE. This lowers binding affinity and releases HSP70 from the polypeptide. The fates of the polypeptide can be a protein is correctly folded, so the job is done, or it might need additional work, so it goes through another round of HSP40 and HSP70 action, or it might need more help, so it's transferred to the chaperone system. and the GRO-EL family, which is present in eukaryotic organelles like mitochondria and chloroplasts, and in all prokaryotes. The structure of GRO-EL is shown, but TRIC is similar. Chaperonins are oligomers with two stacked 7-membered rings that form a barrel shape. The rings interact with GRO-ES, which is a helper protein. The entire chaperonin cycle is shown in the diagram. We will break this model down and go over each step individually. In the first step, the unfolded protein binds to the GRO-EL pocket not blocked by GRO-ES. Then ATP binds to each subunit of the GRO-EL heptamer. After that, ATP hydrolysis leads to release of the 14 ADP and GRO-ES. This is followed by the binding of 7 ATP and GRO-ES to the GRO-EL with the filled pocket. The protein falls inside the enclosure. Then the protein is released in its fully folded shape or in a partially folded state that is committed to adopt a native conformation. If the protein is not folded when released, it is rapidly bound again to another cycle. Molecular chaperones also have a role in protein sorting, which gets a protein to where it is needed for function. Some proteins remain where they are synthesized, but others need to be translocated. Membrane passage issues during sorting can dictate when a protein is folded. Many proteins contain targeting domains which act like address labels. They are usually present unless a protein stays where it's synthesized. These domains are frequently removed at the target site by proteolytics processing. Targeting domains are frequently amino acid sequences but can contain oligosaccharides. Targeting domains are named based on the target location. So we have signal peptide ER, transit peptide chloroplast, vacuolar sorting signal vacuole, nuclear localization signal nucleus, pre-sequence mitochondrion, peroxyzyme targeting signal peroxyzyme. Some proteins contain multiple targeting domains. One example is the enzyme invertase, which has two forms. One is secreted to the cell wall and the other is transported to the vacuole. Both have signal peptides SP and are directed initially to the ER. The vacuolar form has a second target domain, a vacuolar sorting signal, that triggers movement from the ER to the vacuole. Cytoplasmic translated proteins targeted to organelles must get through a membrane. While folded proteins can pass a membrane within a vesicle that fuses with the membrane, other proteins are too big and have to be in an unfolded state if they have to fit through a membrane pore. Since it would not be efficient to fold, then unfold, then refold the protein, it remains unfolded until after membrane passage. This process is assisted by chaperones which keep the polypeptide unfolded until it is through the membrane pore. They can also help attain correct structure after passing through the pore into the organelle. The table shown here shows the 5 major families of heat shock proteins. The HSP100 family can form 6-membered rings that help separate aggregated proteins. While they don't prevent aggregation, they have been described as molecular crowbars that separate small aggregates. Separated proteins can then go through HSP70 assisted folding cycles. The HSP90 family is primarily involved in regulating the heat shock response via interactions with transcription factors or element of signal transduction pathways. They are found in the cytoplasm, chloroplasts, mitochondria, and the ER. The HSP70 family are found in the cytoplasm, ER, chloroplasts, and mitochondria, and they help in the regulation of the heat shock response and stabilizing new proteins. The HSP60 family has two groups. Group 1 is in the plastids in the mitochondria, and Group 2 is in the cytoplasm. The HSP60 family act as chaperoning folders. They may help stabilizing membranes. The small HSP group represents a group of proteins that functions as holders rather than folders. An example on that are the HSP40. That is, they bind proteins until they can interact with the HSPs with the folding roles. They too may stabilize membranes by preventing hyperfluidity. While the heat shock response has been extensively explored, there are many proteins regulated during heat acclimation that do not have molecular chaperone activity. Signal transduction pathways for non-HSP heat acclimations are poorly understood, but studies have shown that potential components of the signal transduction pathway of non-heat shock proteins include ABA, hydrogen peroxide, calcium, ethylene, salicylic acid, protein kinases, and lipid signaling systems. Some of the gene products of these pathways are involved in proteolysis. This is logical based on a buildup of damaged proteins during heat stress. Ubiquitin pathway genes are upregulated during heat stress due to an increased need to remove these damaged proteins. Another mechanism whereby organisms can increase heat tolerance is through accumulation of chemical chaperones to protect protein structure. In yeast, trehalose accumulates and protects cells against high temperatures. Note that the chemical chaperones have a mechanism distinct from molecular chaperones. While the latter are polypeptide binding proteins, effective chemical chaperones are thought to be preferentially excluded from the hydration shell of protected protein. The diagram shows that co-solvent effects on protein thermostability can span a continuum from destabilization to stabilization. Enhanced protein thermostability in the presence of applied mannitol is evident. While some plants can accumulate relatively high levels of chemical chaperones, it is not a universal phenomenon. There may be potential for engineering greater stress tolerance via this route since plants transgenic for glycine betaine had enhanced performance at high temperatures. Activated oxygen defenses have been implicated in heat stress resistance from several perspectives. We will cover activated forms of oxygen in greater detail when we explore chilling stress. For now, it will be sufficient to recognize that oxygen can be made much more reactive than the form we breathe. Whether or not activated oxygen is beneficial or harmful depends in part on the molecular species and amount. Low levels can serve as second messengers in acclimation responses, while elevated levels can overwhelm defenses and tip the scales in favor of oxidative damage. During heat acclimation, chloroplast membranes are stabilized by increasing the digalactosyl diacylglycerol to monogalactosyl diacylglycerol ratio and reducing the proportion of double bonds in the fatty acids. These changes may help prevent hyperfluidity. By the end of the second part of high temperature stress, you should know the following. What is the difference between plant tolerance and avoidance? What are the characteristics of heat shock response? How are heat shock response transcription factors activated? And what are the functions of heat shock proteins? What are the 5 different families of HSPs? And what are the functions of non-HSP? Thank you, and we'll see you in the next module.